In 1964, Supreme Court Justice Potter Stewart faced a difficult legal task, defining the exact boundary of hardcore obscenity. His conclusion was famously brief: "I know it when I see it." Biologists face a similar struggle when trying to pin down the moment a collection of atoms becomes a living organism. While we easily distinguish a dog from a rock, the technical transition from chemistry to biology remains difficult to define. Look closer, and the boundaries dissolve. A virus lacks a metabolism and cannot replicate without a host, yet it carries genetic instructions. Fire consumes fuel and grows, while some RNA molecules can copy themselves without any cellular machinery at all. This ambiguity drives the ambition to build life from scratch in a laboratory. Success here would clarify how inanimate molecules organized into the first cells 4 billion years ago, while offering the tools to manufacture synthetic tissues and blood for emergency medicine. For decades, scientists utilized a top-down approach. They took existing bacteria and methodically deleted genes to find the bare minimum required for survival. Craig Vanter's team eventually stripped a genome down to just 473 essential genes. But removing parts from an existing cell only reveals what is necessary to keep a pre-built machine running. To truly understand the origin of life, researchers must go bottom up, assembling simple, non-living chemicals into a functioning unit. This requires bridging the gap between basic chemical reactions and the complex coordination of a biological machine. At the University of Minnesota, Kate Adamala's team developed a synthetic structure called the spatcell. This 23-micron vessel is assembled from a mixture of lipids and a minimal set of 36 genes. To function, this assembly had to perform the most basic requirements of life, starting with metabolism. The team needed a way to make these synthetic shells absorb nutrients from their environment. They utilized lipid physics. With one water-seeking and one water-avoiding end, they automatically form a spherical shell in liquid, creating a distinct interior world. To eat, researchers added alpha-hemolysin proteins. These embed into the wall like tunnels, allowing nutrients to pass inside. As the cell consumes these nutrients, it grows. Without a way to divide, that growth would eventually cause the membrane to rupture. Natural cells use a complex network of proteins to pinch themselves in half, a mechanism that required billions of years of evolution. Since the spatcell only had 36 genes, the team couldn't replicate that biological complexity. Instead, they used a biophysical workaround. By adding a specific protein that crowded the outside of the membrane, they created enough physical surface pressure to bend the cell inward. The force eventually caused the structure to snap into 2 separate daughter cells. These experiments showed that 2 of the primary functions of life, eating and dividing, can be achieved through the predictable application of physics. The final requirement for a living system is evolution. To be considered part of the biological world, these cells must compete and adapt. The team tested this by creating a mutant spatcell. They gave this variant a gene that produced a more aggressive version of the nutrient tunnels, allowing it to feed much faster than the original cells. They placed an equal number of original and mutant cells into a single environment with a limited food supply. Natural selection took hold immediately. Within 5 generations, the faster-feeding mutants outcompeted their relatives, increasing their share of the population from 50 to 70. By successfully competing for resources and passing on their traits, these chemical assemblies entered the realm of evolutionary biology. This success raises a difficult question. Has science finally crossed the line into creating independent life? There is still a significant gap. Spatcells cannot build their own ribosomes, the internal factories that produce proteins. Without a way to manufacture their own hardware, the cells naturally break down and fail after 5 to 10 generations. This fragility serves as an important safety feature. Because these cells cannot survive long-term outside of a controlled lab environment, the risk of them being modified into persistent environmental or health threats is significantly reduced. This safety is a central part of the debate surrounding biotech, a nonprofit that has open-sourced the team's research. While sharing this data could accelerate the development of new medical treatments, it makes the tools for synthetic biology accessible to those who might use them for harm. Regardless of the risks, the achievement mirrors the Wright brothers' first flight in 1903. That first 12-second flight did not result in a practical way to transport people or cargo across oceans. However, it provided proof that the laws of physics could be used to overcome gravity. The spatsell demonstrates that the transition from chemistry to biology follows a readable mechanical logic. Humanity has moved from observing the mystery of life to learning how to build its most basic components.